This is the picture for the spleen. We have here the capsule of the spleen. It is characterized by being very thick, as you see here. It is formed of the same structure of capsule of lymph node, except that it is rich in smooth muscles. It's very rich in smooth muscles. And also you should remember that it is covered with peritoneum. It is not evident here in the picture. The trabeculae are very thick, and they contain actually blood vessels, trabecular blood vessels, and they arise mainly from the hilum in addition to the undersurface of the capsule. But in case of lymph node, they arise only from the undersurface of the capsule. Here, of course, you know that it has a reticular formation, but it cannot be seen here because it is not, this slide is not stained with silver, and it only appears by silver. So we have only in this capsule from the stroma, in this slide from the stroma, we have the capsule and the trabeculae only, and then we have the parenchyma. The parenchyma is formed of white pulp because it appears white in fresh state, but now it is not white, it is stained because it is formed of lymphocytes, so it is blue, and it is characterized by having arterioles called follicular arterioles or central arterioles. In between these white bulbs, we have this area, which is called the red bulb because it appears red in the fresh state. This is the red bulb because it is red in the fresh state, and this is the trabeculum. These are the white bulbs, trabeculae, and the capsule. Again, in this picture, we have the capsule. Don't forget, it is similar in structure to the lymph node, but more rich in smooth muscle. In lymph node, the smooth muscles are only present at the hilum, and it is also covered by peritoneum, but it is not evident in the picture. Here we have the trabeculae. Here all of these are the trabeculae, and this trabeculum containing blood vessels, and as the wall of the blood vessel is thin, it is a vein, so it is a trabecular vein in this picture. This is the stroma, and then the parenchyma is described as white pulp, which is white in fresh state, formed of lymphatic follicles, and characterized by having these arterioles. These arterioles are called follicular arterioles or central arterioles, these arterioles were absent in the lymph node. So if you are asked, mention one difference that is present in this picture to differentiate the spleen from the lymph node, is that here the lymph follicle have a follicular arteriole which is not present in the lymph follicles of the lymph node. This is as regard the white bulb. Here we have in between the white bulb, which are the follicles, we have red areas. These red areas are called red pulp. So the parenchyma here is formed of white pulp and red pulp, while in the lymph node we have cortex and the medulla. Here we have the spleen also. We have the capsule here, which is red, thick, and then we have the trabeculae, red also, and they are actually mainly arising from the hilum of the spleen, and we have this blue structure, which is the lymph follicle here, called the white pulp. It is characterized by having this follicular arteriole or central arteriole. It is actually not central. It was central at the start. It was in the center, but with the arrangement of the lymphocytes later on, these lymphocytes will be collected at one side, making this arteriole eccentric in position. So this is the cause of the originally central arteriole, and actually the most perfect name is the follicular arteriole because it is now not central no more. Here we have also the red pulp, which is formed of the blood sinusoids and the different types of the blood cells. Here you may be asked about the structure of the red bulb, or what are the cells present in the red bulb and what are the cells present in the white bulb, and that will be discussed shortly. In this slide we will discuss the structure of the white bulb. First of all, this white bulb is formed of rounded or oval nodules. First thing, it is rounded or oval nodules. Each bulb here has an arteriole at one side, which is called the follicular arteriole or central arteriole, which is eccentric in position. So it has an follicular arteriole. Number 3 here, we have 4 different zones. 1, 2, 3, 4. The one which is the marginal zone. يبقى 1 هنا. 1 is the marginal zone. Number 1 here is the marginal zone. This marginal zone, you should study the cells present in the marginal zone. It contains, يبقى number 1 اللي هي marginal zone, it contains the T and P lymphocytes, T و B lymphocytes, و plasma cell, و macrophage and other antigen presenting cells. You should study the cells present in the marginal zone. We have T, we have B lymphocytes, and her daughter, which is the plasma cell, in addition to the assistant macrophage and other antigen presenting cells. This is the marginal zone. Then we have number 2. Number 2 is the follicular zone. This is the follicular zone, this area. The follicular zone, as you see here, it is dark than the germinal center, and it is formed of mainly B lymphocytes. Then we have the germinal center, number 3. Here, this is the germinal center, which is pale or lightly stained, and it is formed of the activated B lymphocytes and her daughter, which is the plasma cell, and her assistant, which is the macrophage. يبقى هنا number 3 is the germinal center, and it contains the same cells present at the germinal center in the lymph node, which are activated B lymphocytes, her daughter plasma cell, her assistant macrophage. Then we have this area, which is the pulse periarteriolar lymphatic cheese. Number 4, periarteriolar lymphatic cheese or pulse. It is the thymus dependent zone because it depends on the thymus in its supply with the T lymphocytes, as it is the area containing T lymphocytes. Of course, in addition to the marginal zone, we have here in the marginal zone, here we have T lymphocytes also. So, if you are asked what are the zones containing T lymphocytes, mainly the thymus dependent zone or periarteriolar lymphatic cheese, and in addition to the marginal zone because it contains also some T lymphocytes. Finally, we have here the follicular arteriole. Here we have a follicular arteriole which is present at one side of the follicle, and it is surrounded with the pulse. Also, we have this area around the white bulb, which will be the red bulb. Here we have the red bulb. This, the white area, compact white area here, it is a trabeculum. Before leaving this slide, we should discuss the structure of the red bulb because you may be asked about the cells in the red bulb. Here we have what are the components of. Here we have to talk about the structure of the red pulp. The red pulp here is formed of blood sinusoids or and splenic cords. The splenic cords has another name, which is Bell-Roth cords. You have to know the name of the cells present in these two compartments. In case of the blood sinusoids, you have to know the cells present in the blood sinusoid. Here in the blood sinusoid in the spleen is lined by the fenestrated cells called staph cells. These cells are characterized also by having a wide intercellular space. So they have wide spaces in between, and they have non-continuous basement membrane. So this is important for the structure of the blood sinusoids. Not only that, but also remember that they have littoral cells. They have littoral cells in and around their vicinity, in around the blood sinusoids. So we have also littoral cells. In and around the blood sinusoids. As regards the splenic cords or the Bell-Roth cords, the Bell-Roth cords here are characterized by having the following cells. They have all the types of the blood cells. They have RBCs, they have platelets, and they have the granulocytes, and they have the non-granulocytes as the monocytes and lymphocytes, hair daughter, plasma cell, hair assistant, the macrophage. So the cells present in the Bell-Roth cords or the splenic cords are the RBCs, here, the platelets, the granulocytes, and the non-granulocytes as the monocytes and lymphocytes. Lymphocytes, you will remember the daughter plasma cell and the assistant, the macrophage, so that these are the cells present in the Bell-Roth cords or the splenic cords. Also, don't forget the functions of the spleen. These functions can be summarized as 4 functions for the blood, which are formation of the blood elements during fetal life, storage of the blood, filtration of the blood, and destruction of the old RBCs. And we have the 5th one, which is the immunological function through T and B lymphocytes. Also, the reticular cells of the stroma can trap the antigen and present it to the lymphocytes. So it has both humoral and cell-mediated immunity. This is a picture for the thymus. Here the thymus is formed of the lobules, as you see. These lobules are separated from each other by the connective tissue trabeculae, which are destroyed in this picture and appear as empty white spaces. In each lobule, you can see it is formed of a dark cortex, and it is blue in color, and a paler medulla in the center, which tends to be of a red color. Why we have a dark cortex and a pale medulla? In the cortex we have the lymphoblast, and they have large dark nuclei, and these lymphoblasts will give rise also in the deep part of the cortex to the lymphocytes. But in case of the medulla, we have lymphocytes, but they are separated from each other. They are not condensed. They are separated by two cells, the reticular cells and the macrophage, epithelial reticular cells and the macrophages. This is all about this picture, the connective tissue septa, the lobules. No more details here can be seen. In this picture, we have the capsule of the thymus here. It is present. This is a capsule on the outer surface. And this we have the trabeculae. The trabeculae here are described to be incomplete. Why? Because it does not separate these lobules completely from each other. We have fusion between the medulla of these two or three lobules. So no complete separation of the lobules from each other. So they are described as thin, incomplete trabeculae. Thin, incomplete trabeculae. This is the character of the trabeculae in the thymus. It incompletely subdivides the gland into lobes and large lobes and lobules. Of course, you cannot see the reticular formation because it should be stained by silver. Here we have in the lobes, we have the dark cortex formed of the lymphocytes and the lymphoblast. We have the paler medulla formed of the lymphocytes, but they are separated from each other by two cells, macrophage and epithelial reticular cells. That's all about this picture. This picture is very nice, demonstrating the characteristic structure present in the medulla, which is called the Hassel corpuscle. The Hassel corpuscle, it is present only in the medulla of the thymic lobules, and it is formed of a central acidophilic mass. This central acidophilic mass, it is a hyaline mass of the degenerated epithelial reticular cells. And this mass is surrounded by concentric layers, layers having same center, concentric layers of these epithelial reticular cells. Don't forget that the thymus has two origins. It has a mesodermal origin for the lymphocytes, and it has an endodermal origin for the epithelial reticular cells. So you may be asked, what is the origin of these epithelial reticular cells? It is endodermal in origin. What is the origin of the lymphocytes? It is mesodermal in origin. Here you can see these epithelial reticular cells. You may be asked about the prominent features you see here. The cells here have large oval pale nucleus with a prominent nucleolus. This is the feature which is apparent here in the picture. You may be asked about the functions. These cells are the nurse cells for the lymphocytes during their differentiation. Also, they secrete thymic hormones which help or promote the T cell differentiation and proliferation. Don't forget, you may be asked about the special features of the thymus. Here the special features are: number one, the reticular cells here are endodermal and not mesodermal. Reticular cells are endodermal, not mesodermal like those of the stroma, and they do not produce the reticular fibers. Number two, it contains these Hassel corpuscles in the picture. Number three, it has no nodules. No nodules here. We have cortex and the medulla. No large lymphocytes, no B lymphocytes, no plasma cells, and no afferent lymphatic vessels. No large lymphocytes, no large lymphocytes. no B lymphocytes, no plasma cell, no afferent lymphatic. So these are the special features for the thymus.